Well, hello everyone, and welcome to today's webinar hosted by Precisionary Instruments. I'm really thrilled to have all of you join us as we dive into some fascinating research at the intersection of plant cryopreservation and advanced imaging techniques. And so our guest speaker today is Heidi Kreckel, who is a PhD candidate at Colorado State University in Nancy Levinger's lab. And actually, Dr. Levinger is also here today. And Heidi specializes in plant cryopreservation and has pioneered the use of nonlinear microscopy to study cryoprotectant interactions with plant shoot tips. She was selected as a CSU Vice President for Research Fellow for her impressive thesis presentation, 3-minute thesis presentation. And I just also wanted to say that she's going to share with us a little bit of the story of how she got into this field of research and also how she's using the vibrating microtome with it as well. So, but more importantly, in today's session, Heidi will be sharing her research on tracking cryoprotectant permeation into living peppermint shoot tips and how the Compresstome vibratome played a crucial role in prepping the plant tissues for high-resolution microscopy. So we do encourage you to ask questions throughout the webinar. Go ahead and make use of the chat box on the bottom. During the webinar, I'll also moderate that chat box, and at the end, we'll have an open Q&A session. So now, without much further ado, please join me in welcoming Heidi Kreckel.
Thank you so much, Abby, and thank you for inviting me. So today I'm going to be talking about some fundamentals of plant cryopreservation and tracking the permeation of cryoprotectants into living plant or living shoot tip sections. But I want to start with a brief talk overview as we'll be navigating quite a bit of ground or information today. So really, I'm going to begin with an introduction of plant cryopreservation and why we actually should care about cryopreservation and why it's become incredibly important. And then I'm going to talk about some challenges of preparing shoot tips for microscopy experiments. And this is really where the Compresstome has come in and helped us be more innovative and more progressive in our research. And I'm going to give a brief overview of some non-invasive microscopy techniques and then go over some of our key study results. So about 40% of the world's plants are considered endangered or at risk at this time. And this is incredibly concerning to us as plant genetic diversity is incredibly important for our global food supply and our agricultural industries, our pharmaceutical industry, and also, of course, your conservation efforts. And many folks in the audience might be familiar with several different conservation efforts that might exist for plants, particularly seed banks, which are considered one of the most cost-effective solutions. However, while seed banks are super helpful and innovative and useful for preserving many plant species, they're not considered a universal solution. And the reason being is shown by these three very common agricultural crops, which are shown here on the left-hand side of my screen. And what all three of these crops have in common is that they actually cannot be preserved via their seeds, so they are known as exceptional species. Now, the reasons why they cannot be preserved vary from species to species. For example, avocado seeds are too large and moist. Bananas only produce non-viable seeds, so of course we cannot preserve those and hope to regrow a banana plant. And lastly, in the case of apples, the apple seeds do not preserve the original genome of the parent plant. In fact, they're heterozygous. And so cryopreservation in recent years has emerged as a potential universal solution to this problem across the board. However, for the folks who are not familiar, cryopreservation is the preservation of cells and tissues at liquid nitrogen temperatures. This is about negative 200 degrees Celsius, which is extremely cold. And so cryopreservation does require a series of steps. It's not as simple as, say, taking a tomato plant at room temperature and then sticking it into the freezing cold temperatures. So, for example, if I stuck a tomato plant outside in Colorado temperatures today, that tomato plant would die as there would be the formation of dangerous ice crystals within the cells, which would ultimately kill them and then kill the plant. And so to help facilitate successful cryopreservation, despite the many different methods, we usually utilize these things known as cryoprotectant agents, also known as CPAs. I'll use those interchangeably throughout the course of the talk, which help prevent the formation of these dangerous ice crystals and thereby help keep our plants alive during the process. Now, in the plant cryopreservation world, we do have a series of cryoprotectant agents that have been used in this formulation known as plant vitrification solution 2 or PVS2 over the past 30 years. In fact, the solution has not really changed, and it comprises of various concentrations of glycerol, DMSO, ethylene glycol, and sucrose. And while this formulation has not changed over several years, it works in some cases, but it doesn't work in other cases. And so really the question that remains is why does the solution work in some cases, but not others? And so what we're trying to do in the cryopreservation group or in the Levinger Lab at CSU is to understand how PVS2 actually works to help support or successfully cryopreserve living cells and tissues. And where we're starting is trying to understand where dimethyl sulfoxide, ethylene glycol, and glycerol in particular go within living cells and tissues. And hopefully by understanding where these molecules go and localize, we can understand better how they facilitate and work within the cell to help prevent that ice formation individually, and also how that behavior changes once we put them in this entire PBS2 solution. And this project was started about 2 years before I joined the Levinger Lab by Dr. Samuels. And this project really started by just looking at rice cells. However, as much as rice cells are really important, they're a great model system. They help us understand the individual cellular basis of what these molecules do. In plant cryopreservation, shoot tips are actually what are considered the best candidates for this particular form of preservation. And so most studies actually work with shoot tips. And so really 5 years ago, I took the next steps in this project by being willing to take on the complexity of working with plant shoot tips. In this case, this is a peppermint shoot tip, which is also shown on what we call a notosection here. However, working with shoot tips is a bit complicated and quite difficult to do. In fact, whole shoot tip excision, which has been cited by many different resources, I've shown one here, requires several months and years of training by experienced practitioners. And what that means or why that's the case is really demonstrated by this photo here, which is the first good hand exercise shoot tip I obtained after about 2 months of practice. And as you can look here by the scale bar and at the overall size of this, the shoot tip is about 150 microns in diameter, which means that this is about in total the width of a human hair. And so in order to excise these as a practitioner, you're working under a dissecting scope, you're excising them by hand. So they look like a speck. And then you're taking this tissue, which is incredibly sticky, and you're hoping to transfer it onto a microscope slide without damaging it, turning it into essentially a paste, if you will. And then on top of it all, your hope is to obtain the desired orientation shown here. And what I mean by desired orientation in shoot tips is shown in this schematic here. Shoot tips essentially comprise of 2 different regions, the leaf primordium and the meristematic region. And all these regions consist of is undifferentiated or rapidly dividing cells, which can form a new plant. So they're the stem cells of your shoot tip. The only difference between them is the meristem contains completely undifferentiated cells, whereas your leaf primordium, these cells typically form new leaves. But even when we can get these perfect orientation, we can transfer these to the slides, our ideal samples still suffer from various issues. For example, the first one would be variation in size or thickness. So if I draw your attention to this diagram here on the right, this graph, we've got our tissue thickness on the x-axis and our mean pixel intensity on our y-axis, which is essentially the brightness that we see in images under microscopy. And the bottom graph and the top graph represent 2 different shoot tips that were hand excised and then collected data from. And as we can see in this top graph, our shoot tips were only about 33 microns thick, whereas in our bottom graph, our shoot tip in this case was about 57 microns thick. And the difference here is shown in the amount of pixel intensity. We see that we only get a maximum pixel intensity on this bottom graph at about maybe 1700, where in this top graph, we're actually able to obtain a maximum pixel intensity closer to 3000. Which means you can see how thickness actually decreases the amount of intensity of light that is able to travel through. We also suffer from this case of lower resolution, which makes it difficult to identify individual cells. And then it's shown in this bright field microscopy or light microscopy image here on the right, where again, we have our leaf primordia regions and our meristematic region here. And as we can see, it's quite blurry in the center, it's quite dark, which means we aren't getting a lot of light through our sample. And we can start to distinguish some individual cells on the periphery here. However, it is almost impossible to actually see within the individual cells, as a whole. And so ultimately, after a great deal of frustration with working with these samples, even when I was able to get these ideal samples, I was only getting a success rate of maybe about 10%. So cutting the samples, cutting them out, transferring the slides, just to get 3 samples would take a matter of hours. And so I thought there has to be a better way. And I was familiar with microtomes from my time doing biology research and biology classes. And so I stumbled across vibrating microtomes during this time, and realized that this could be a potential way to actually expedite and better modify our ability to create good samples for microscopy experiments. And so I was extremely lucky to obtain access initially to a Compresstome, vibrating microtome at the University of Colorado Anschutz Medical Campus in Dr. Richard Benninger's laboratory. And he was nice enough to allow me to come down, learn how to use it, which is shown here. This was actually the laboratory manager, Mara Sicko Evans, showing me how the Compresstome actually worked. And so after I was trained on that, on the vibrating microtome, I then really began the task of optimizing it for slicing around samples. However, after doing a lot of literature research, I found out that very few research groups have actually used vibrating microtomes to section shoot tips as a whole. And so I was very much starting from scratch in terms of how to use this instrumentation to prepare these shoot tips for microscopy experiments. And so my very first challenge actually began very, very early on in the very initial stages where I started by mounting the sample. And as you can see here, I have a nodal section with different shoe tips. It's kind of hard to see. There are little dots on either end of that nodal section. And as you do for most vibrating microtomes, is you mount the sample perfectly flat onto a sample stage with just some super glue. And when I did that, it didn't quite work out, as the vibrating microtome, particularly the Compresstome, it has a built-in feature which prevents you from slicing too close to the sample stage in order to protect the integrity of the sample stage, which is great. It's not a huge issue usually. However, in our case, our samples are so small and so thin that this actually fell within that range. And so we weren't even able to slice our sample at all upon the initial attempts. And so that form of mounting was immediately out the window and not a way that we could prepare samples. And so the next question was, how do we elevate our sample to get it off that stage and outside of that window that precludes us from actually sectioning? And the first answers that came to me was really through the use of these 0.25 millimeter thick sticky spacers. For folks who are unfamiliar with our research, essentially we create a series of calibration chambers to collect images for various concentrations of these cryoprotecting agents that we're working with. And to do this, we essentially have these nice spacers with these 4 chambers that we then pull these little circular spacers out, and typically we would just throw them in the trash. However, I thought, okay, why not, instead of throwing these in the trash, why don't we actually stack them on top of each other and create a little spacer that we can put down and then mount our sample in? The next step was really identifying what type of super glue to use. In a lot of cases, especially if you're working with mammalian tissues, regular liquid super glue would be totally fine. However, in our case, gel super glue has really made a massive difference. And that is because gel super glue provides additional stability and support for our stem when we place it into the different super glue. What it also enables us to do is, once it has gotten about halfway dry, it allows us to adjust the angle at which our stem is in the glue and also rotate our stem to obtain an optimal configuration or orientation for slicing. And so these 2 components really made a big difference in the initial steps in order to mount our sample accordingly. Once I identified how we could actually raise our sample off the stage, the next question became, okay, do we mount our sample vertically, or do we mount our sample horizontally at a different angle? And really the answers to this question came quite quick. And that comes down to, again, if I point this nodal section here, where again I have shoe tip here and a shoe tip here. If I were to mount my sample vertically, as shown here, essentially the blade would have to slice across my shoe tips as shown by this arrow here. Now, what I believe is quite obvious in this case is that as we were to slice our sample here, we are much more likely to cause greater damage to our sample, potentially lose this shoe tip entirely. And so this immediately became not an option we wanted to pursue. However, if I were to look at this sample on the right here, if I mount it horizontally at an angle, my blade is still able to travel upwards across our sample, which then allows us to reduce the amount of damage at slicing our shoe tips and potentially enable us to collect multiple shoe tips on the same slice. And so we immediately rejected this vertical orientation and then switched to mounting our sample at this horizontal orientation at a slight angle. But another issue actually arose fairly early on, which came to be the mounting or the orientation as we placed our sample into the vibrating microtome. So if I look at my shoe tip here, as you can see, the base is mounted towards the bottom, but our blade would be traveling through a sample and hitting the top of the sample first. And what I found actually occurred oftentimes when I oriented my sample like this in the vibrating microtome is that the blade would actually shear or almost completely remove my sample from the actual mount, and it would completely destroy the integrity of the sample. And so at the end of the day, it really came down to the proper orientation is we wanted to mount it so that our sample with the super glue and the sticky spacers holding the stem was closest to the blade as it traveled through our sample. And then we had the added benefit of with the angle of our sample being upwards and our blade traveling down, you kind of have that opposite force pushing back against that blade, which actually improved the integrity of our samples as we slice through. And so once we optimized the actual sample orientation and mounting, we then had to optimize the typical parameters. So for folks who are, of course, familiar with vibrating microtomes, you have various parameters you need to adjust, such as agarose concentration, and then the actual instrument settings themselves, which in the case of the Compresstome would be the advance of the speed and the oscillation of your blade. And in our case, the nice part is I did have a starting point. I was able to access the Compresstome via 5100Z manual and see the starting point in terms of the suggested cutting parameters for plant leaves, stems, and seeds. And because leaves and stems were closest to the tissue type that we were working with, I began by investigating the advance in speed around 3 to 5 and oscillation around 4 to 6, as well as the suggested agarose concentration at about 2%. And what I found is that an agarose concentration of about 2 to 4% worked best. 2% if you wanted to have easier access at removing your sample from the agarose. For example, in this right-hand image, which is my first successful sample at 100 micron stick, we can see some of that agarose in the periphery here. However, 4% was really well if our plant had grown out a little bit more, was less supported by the stem. For example, you can see that my shoe tip is quite close to the stem here. It's super supported. And so 4% helps a bit with that. In terms of the instrument settings, we optimized them to find that an advance or a speed of 4 worked best for this type of tissue, as well as a blade oscillation of 5. However, in our case, we are also, again, thinking in terms of microscopy experiments. We want to be able to take images of these samples, and that means that we want to make sure that our thickness is as thin as humanly possible so that we can actually see as much as possible as we're looking at our samples. And so we looked into and optimized the thickness and found that 50 micron thick sections were the best thickness that we could go with. I did attempt to go lower in terms of thickness. However, anything below 50 microns is about when we would start to see more variation in sample thickness, or we would have issues with actually just shearing our sample out of the encasing altogether. But what we did find with 50 micron thick sections over 100 micron thick sections is that we had far greater success with actually sectioning these and coming up with great samples, such as the one shown here on the right, and that these were much more ideal in terms of thickness for our microscopy experiments. And this is supported by literature measurements, which also demonstrate that tissue thickness for plants that are up to about 50 microns are typically ideal for these types of experiments. And lastly, we also needed to confirm our sample viability. Again, we are working with tissue that we hope is alive, so that we can actually do live tissue experiments for measuring these dynamics in real time. And so we did that, or I should say, my colleague Albert Ochoa Castillo, who was nice enough to do this for me, conducted these Evans blue tissue staining experiments to confirm the sample viability. If you're unfamiliar with Evans blue works by permeating cells that are dead and not permeating cells that are alive. And so if we look at this image here, this is a section shoe tip under light microscopy before it was stained with Evans blue. But in this central image here, we are looking at the same shoe tip after it has been stained with Evans blue. And well, if we look at this, it's looking a bit dire. This outer layer looks extremely dead. We can see it's filled with this dark blue coloration. However, because we are slicing, the top layer of these cells are likely dead because we just sent a blade through them, and our bottom layer of these cells are likely dead as well. And so I had faith, partially because we can also see this little bit of opening or this brightness in this region here, which could indicate that these central layers are actually alive. And so to investigate this, Albert went in and conducted some additional fluorescence microscopy experimentation, which is this image here. And as we can see, is Evans blue is only fluorescing in areas surrounding the cells, or what we would call the apoplast, which indicates to us that these central regions are in fact alive, and that they do remain alive for about 24 to 36 hours following sectioning. I do have to note that after about 48 hours, our samples are no longer alive after sectioning. However, it was important for us to find out that our initial experiments being run within about 24 hours of sectioning are being done on live shoe tip sections. And so to give an overview of all the different challenges that we addressed, our sample mounting or orientation is extremely key, and that is demonstrated in this figure here on the right, where as we can see, we mount with the base in super glue on these sticky spacers and at an angle, and then we slice from the base towards the top. We also determined that agarose concentration of 2 to 4% is ideal, as well as the instrumentations of advance or speed of 4 and an oscillation of the blade at 5 work best for sectioning. Our optimal sample thickness is about 50 microns, and our samples remain viable for about 24 to 36 hours after sectioning. However, at one point in this project, I did lose access to the vibrating microtome that I was using at the University of Colorado Onship Medical Campus due to the fact that I just couldn't get in contact with folks anymore. And so we really tried to shift our experiments to another vibrating microtome on Colorado State University's campus. And that vibrating microtome happened to be a traditional vibrating microtome where your mounting stage happens to be in a horizontal plane, and it was a Leica version to be exact. And what we found is that sample preservation or preparation in this method is really only successful using Compresstome models with that particular technology where you're slicing a sample that is held within a cannula horizontally, or I should say vertically, towards the blade. And essentially, the traditional vibrating microtome dislodges samples from our horizontal stage. In fact, when my colleagues Kylie and I, we were trying to cut these, we would be like putting walls of super glue up on the other side of the agarose mount, and it would still be just shoving the agarose mount completely off the stage as it was sectioning. The other thing that we found is when it did happen to remain mobile or remain immobile and actually slice, is that we had an incredibly inconsistent sample thickness across the board, even when we were sectioning at 100 microns. And this is shown in the top part of this figure here, where we can see that all of these were set at 100 microns, but we can see that, for example, from this sample to this sample, our thicknesses vary massively between the two. And then in addition to that, we also saw a huge amount of increase in damage due to lack of our sample stability. And that is just shown across the board in this top part of this figure here. We can see that there's a lot of damage, a lot of debris, in particular across all of these samples, which indicate that the sectioning was not nearly as clean and precise versus 100 micron slices obtained using Compresstome models, where we can see, again, our samples are much cleaner, our thicknesses are much more uniform across the board. And so really, at the end of the day, as much as this methodology does work, it really only works using this technology that is encased by the Compresstome models. And I do have to note that this type of sample preparation has hugely impacted or benefited our experiments by addressing several of our challenges that come with whole shoe tip excision. For example, again, I mentioned at the very beginning, especially with this image here on the left, this bright field image, that we don't have a lot of control over our sample thicknesses. And we now have much more control in that all of our samples are 50 microns thick. We also can see clearer cell layers. Again, looking at this image here on the left, this bright field image, and then this fluorescence microscopy image here on the bottom. We can see that we don't see as clear cell layers. It's much thicker. Whereas if we look at these images of another section shoe tip on the right, we can see that now we can see these almost clear cell layers. We have much more uniformity in terms of our thickness. And so we have that increased resolution in order to do much more advanced image analysis following our experiments. The other two things that the Compresstome has enabled us to improve upon is our training time. Again, for hand excised shoe tips, that requires a lot of training and experienced practitioners. And that usually takes on the order of months, if not years, to obtain that experience. Whereas in terms of using a vibrating microtome, we've actually significantly reduced our training time down to a matter of weeks for new practitioners. And lastly, because we're able to control our sample orientation during sectioning, particularly with that nodal section where we can rotate it or adjust the angle, we no longer have to depend largely upon luck to obtain this desired sample orientation. That is now something we have a ton of control over from sample to sample. And so now at this point, I've gone over an introduction to plant cryopreservation and why we should care about it. And I've discussed some of the challenges of preparing the shoe tips for microscopy experiments and really how the Compresstome has helped advance or improve upon our ability to prepare good samples for microscopy experiments. And so now at this point, I want to remind everybody that again, the overall focus of the Levinger Lab is to track where cryoprotecting agents, that being the components of plant vitrification solution 2, go within living plant cells and tissues. And we want to do this using non-invasive techniques. And so this is going to transition into the second half of my talk, which is going to go over these non-invasive microscopy techniques that we use and some of our key study results. And so our overall project design, which I want to start with, is we essentially take these cryoprotecting agents, again, that being DMSO, glycerol, or ethylene glycol. We place them on the edge of this perfusion chamber here that we make, which comprises of silicone grease and a slide cover slip and some media, as well as a contact wipe to essentially induce that flow. We put that on the edge, and then we're able to flow that over our shoe tip and then collect microscopy data before and after that flow to see how things change. And we collect essentially 3 types of microscopy data simultaneously using a Olympus microscope that is located at the University of Colorado Denver Onshore Medical Campus, which is depicted in this diagram here, where we essentially are able to collect 3 different types of data, that being FAD endogenous autofluorescence, which is essentially a metabolic molecule that exists within living samples. We also are able to collect chlorophyll endogenous autofluorescence, which is, of course, the center of photosynthesis for most plants. And lastly, we collect data from this type of microscopy known as coherent anti-Stokes Raman scattering microscopy. Now, to give a brief overview about what coherent anti-Stokes Raman scattering microscopy is essentially a form of Raman microscopy. And for folks who are unfamiliar with Raman microscopy measures different molecular vibrations. So if I draw your attention to this water molecule here, we can see that it's moving in all these different ways, and then we can see different frequencies that are shown in this upper left-hand corner. Now, if I were to look at an actual spectra of what this would look like, or a Raman spectrum, I want to draw your attention to this green trace, which is shown here on this Raman graph on the right, where we have our frequency again on the x-axis and our normalized intensity on the y-axis. And what we can see here is that this peak ranging from about 300 to about 3,700 wave numbers would actually be all of the frequencies of this water molecule's vibrations that we're seeing here on the left. And so each of these various peaks do represent all the potential molecular vibrations that occur within this living cell. Now, in terms of CARS microscopy, what this type of microscopy is doing, and all you really need to know about it, is that it's used to track a single chemical bond of interest. So essentially just one of those bonds motions. So for example, if we have a CH bond, such as the one in dimethyl sulfoxide, where we're shown by the CH3 molecule, or CH3 here, essentially that bond moves at a frequency, which is demonstrated by this black trace here, at about 2900 wave numbers. Okay, and as you can see, this is occurring under another peak in that rice cell spectrum, in the green peak. And this is not surprising to us, as rice cells or a lot of proteins and lipids contain these CH vibrations within your cells. And so what's unique about coherent anti-spectral scattering microscopy is by being able to track a single chemical bond of interest is we can actually change out this hydrogen with a heavier form of hydrogen known as deuterium, which then moves this vibration down to a lower frequency right here at about 2100 wave numbers in this vibrationally quiet region of that plant cell spectrum that is shown in green, and now allows us to just detect that carbon deuterium or that CD bond vibration that is occurring in that region. And so this is particularly beneficial to us over, for example, folks, if you're here and familiar with fluorescence tagging, this is particularly beneficial to us as adding a deuterium molecule to these small osmolates is very similar to me simply walking around my house with my cat on my shoulder, as depicted here. It doesn't impact my ability to move around that much, and essentially allows me to access all different corners of my house or small rooms. And that would be essentially what this carbon deuterium bond allows us to do, is not only track these molecules in real time and only obtain signal from them, but we can also track them without impeding their motion as they travel throughout the cell and even into small compartments within those cells themselves. And so at this point, let's talk about some of the data that we've collected over time. I've collected data for all the different components in plant vitrification solution 2, save that as sucrose. The reason why we haven't collected, or I should say I haven't collected data for sucrose, is up until recently, we haven't been able to synthesize it, deuterated sucrose. However, my colleague, Albert Ochoa Castillo, is now able to synthesize deuterated sucrose, and so that is really where his project comes into play. However, on my end, I focused on ethylene glycol, DMSO, and glycerol. And as of this point in time, I've collected data for both DMSO in media and PBS2, ethylene glycol also in media and PBS2, and glycerol in media and PBS2, meaning all these components on their own versus how they change once they get into this overall PBS2 solution shown here in this upper right corner. And again, to remind folks, we've collected 3 different types of microscopy data simultaneously, that being FAD autofluorescence, essentially a metabolic molecule within our samples, chlorophyll autofluorescence, which is again a photosynthetic molecule within our plant, and lastly, the CARS, or coherent anti-Stokes Raman scattering data that I just discussed with everybody. And for the sake of time, I'm just going to focus on 2 different molecules, which is DMSO and glycerol, and the data associated with each of them. And so starting with DMSO, I'm going to go over DMSO in media, so essentially how it behaves on its own versus how it behaves all together with the other molecules in PBS2. And the types of data, and this is what it's going to look like across the board for all of the remaining slides of this presentation, is we're going to show a autofluorescence composite image. And a composite image simply means that I collected 2 images and I stacked them on top of each other. And this composite image is of chlorophyll, which is in magenta, which we like to see that there's not a bunch of that here. And then FAD, which is in green. So that's what we can see the majority of our sample in. Now, the unique part about FAD autofluorescence is we can start to see some individual cells. And so that's what we're going to be using to identify individual cells throughout the remainder of this talk. Now, on the right-hand side of this image, we're looking now at a CARS image, where we have a Viridis color scheme applied from purple, which indicates little to no DMSO, or where light has been blocked, versus yellow, which indicates higher concentrations of DMSO, in this case 19% weight by volume. I want to direct folks up to the top here and demonstrate or explain that we are only actually dosing with 15% weight by volume DMSO. So higher concentrations, or where we're seeing these higher concentrations of yellow, indicate to us there could be potential pooling. Now, the major results that we see here is that DMSO, through the color changes, is actually permeating quite deeply into all regions of our shoot tip section here. We can also see through the variation that it's not uniformly distributed throughout, and that we are seeing concentrations, again, as high as 19%, which again equals pooling. Now, as I mentioned on the left here, FAD autofluorescence allows us to identify individual cells. And it's important to note that in shoot tip samples, particularly meristematic cells, nuclei comprise of about 90% of our cell volume. So when I identify individual cells, I'm not just identifying individual cells, I'm identifying nuclei in large part of that volume. And so that's really the data that we're interested in obtaining, is how these molecules impact our nuclei and what concentrations are getting in. And so what we found when I identified these individual cells, if not these nuclei, is that we're seeing about 7% permeation of DMSO within, when it is simply acting on its own within media. Now, when we switch and put DMSO within PBS2, what we see is that our permeation seems to increase on the right here. We have much more higher volume of color that is occurring across all of our regions in our sample. We see greater concentrations of yellow, in this case, that being about 16% weight by volume, which is now closer to the dosing concentration we're working with. And we can see that it's getting in, and it also appears to, well, still not being uniformly distributed, to be more uniformly distributed than before. And again, if we isolate individual cells or regions within the meristematic region here, we have now identified that our concentration of DMSO that is now permeated into these regions has increased by a couple of percent, up to 9.2% weight by volume. Shifting into our glycerol data, which is arguably our more interesting data for this particular area. In media, we do find out, again, we are dosing with 30% weight by volume glycerol, shown up here. And when we look at our CARS image on the right, we can see that our glycerol is still permeating into all regions of our shoe tip. However, it doesn't appear to be permeating a lot. Again, on this scale bar, we have in purple little to no concentrations of glycerol, and in yellow higher concentrations of glycerol. And what we can see is that our permeation appears to be roughly around 18 to 20% within these deep regions. However, we do still see some pooling occurring within our samples, and we do see that deep permeation. And then again, to look at some of the data within the individual cells, if not the nuclei by extension, we do record that we see about 17.1 weight percent, or weight by volume percentage of glycerol getting within our sample. However, again, when we put it in PBS2, we see that behavior change, and in a way that is quite significant. In this case, now we see that we have low concentrations ranging from 0% weight by volume glycerol to actually upwards of 58%, which is almost double the concentration of glycerol that we're dosing our samples with. And as we can see, is that concentration is increased considerably within all of these central regions. We see a much higher permeation, if not pooling at these concentrations within our cells of our sample, which not only indicates that the overall PBS2 solution is now permitting glycerol to permeate much more deeply into our sample, but that we are now seeing a preferential accumulation of glycerol within our cells. And again, to confirm what we're seeing, we can identify individual cells, and we have noted that that concentration within these individual cells has indeed increased, if not doubled, to about 29% weight by volume, which is now matching the concentration of glycerol that we are dosing our samples with. And so overall in this project, what we found is that our cryoprotecting agents, that being glycerol, dimethyl sulfoxide, ethylene glycol, all better permeate our tissue when they are acting within this overall PBS2 solution versus when they are by themselves. We have also found that DMSO and ethylene glycol behave very similarly in that they have more uniform accumulation within tissue while they are in PBS2. We also see a slight increase in the nuclear concentration for DMSO from about 7% to 9.2% weight by volume. Now, in terms of glycerol, we do see an increased average concentration in pooling within tissue as well from what we are seeing within PBS2. And we see our nuclear concentrations almost double from about 17% to 29%. So really, what does this all mean for us, and why should we care about these concentrations? To start with DMSO, DMSO is actually considered quite toxic. For example, in a lot of different studies out there, particularly in mammalian tissue, it is known to cause issues with nuclei. It's known to cause potential problems in terms of the integrity of membranes. And so GMSO is something we really want to make sure is not causing any potential toxic effects against our cells. And so in terms of DMSO, the literature measurements are typically recorded in volume by volume. And so by essentially converting these weight by volume measurements, volume by volume, we then find out that some particularly concerning information. Now I want to note that our concentrations increased from 5.9% volume by volume to 7.7% volume by volume PBS2, and then these are minimum concentrations. Again, we are collecting this data from light that is permeating through 50 micron thick tissue, which means we are losing some of that. Light intensity as it travels through our tissue, which means when we collect our calibrations, we cannot account necessarily for the decrease in light, which means these are minimum concentration estimates. And so where this gets concerning is if you look into literature, plant microtubule polymerization, which essentially affects our nuclei and reproduction processes within cells, is that this polymerization occurs anywhere between 8.0 and 12.0% volume by volume, which means 7.7% volume by volume is eerily close to this, which provides us with potential implications for cell damage at these DMSO concentrations within our nuclei. Now, in terms of the permeation in PBS2, some of the other interesting results that we have is that DMSO and ethylene glycol concentrations range typically between 14 and 16% weight by volume, and these appear to match our dosing concentrations at about 15%, which means to us that these molecules are essentially perfusing or diffusing back and forth across our membranes pretty equally across the board. However, because we see our glycerol concentrations almost double the original dosing concentration, we are now seeing some preferential accumulation within our cells, and that still remains something that we are trying to explain as of this day. And lastly, probably the most interesting component that comes in with this glycerol data and result is that this accumulation does match the natural physiological concentrations that we observe within some cold hardy organisms, for example, insects. So while we have yet to thoroughly understand exactly why glycerol is accumulating within these cells, we do know that these concentrations could be a reason why PBS2 is considered to be so effective for plant cryopreservation. And so on that note, I want to thank all my funding sources, the various user facilities I have used to access this instrumentation and collect this data. I'd like to thank my lab group for all of their help and support, as well as Dr. Gail Boak, Dr. Dominic Stitch, and then Dr. Fiona Samuels, as without them, this project would not be a success. Thank you.
Heidi, thank you so much. This was fascinating, and the images that you have are really beautiful. And you troubleshooted with sticky spacers, which I think our company will start implementing to solve that issue. And believe me, that's commonly encountered just because, we don't want the blade to hit the specimen tube for the vibratome. Thank you so much for your talk. I do want to open up the floor to our attendees. So those of you who are here, like Marco, Roxana, Stephanie, if you wanted to ask questions and unmute yourself, please feel free to do so. And just giving a moment for everybody to kind of, unmute and chime in if they want to. But if not, and you wanted to communicate with the future Dr. Kreckel, please reach out to me, and I'm happy to put you in touch with her. So on that note, I just wanted to send a big thanks to you, Heidi. Good luck with your graduate studies, and also a thank you to Nancy Levinger here for her lab.
Thank you for having me.
Wonderful. Everyone have a wonderful week. Take care.
You too.